Hello.
Hi, Vidhi.
Hi, how are you?
Hi.
Hi.
Hi, Vidhi.
Hi.
Hi, Lalith.
Okay, welcome. So who wants to do their update first?
Anything is fine for me. Anything works. Right. So continuing with the project. So so week 11. This is week 10, and it was about continuing with the UI. So right. So in the UI, I have added 2 new features, and the 2 new features are dropouts per step and tasks per step. So earlier, these features were like manually hard coded in the code itself, and you couldn't adjust it in the code. So now, in the UI, but now we have the feature in the UI to adjust these two. So we have dropouts per step and tasks per step. So manually we can adjust. So earlier there were like the dropouts per step were like 2 agents dropping out per step, and the tasks per step were set as 3. So let's see what the benefits or changes are that I have observed. So that it obviously offers dynamic control because the UI will offer the user to control it as per the needs. So it's not necessary that in every step 2 are dropping out. Like the number of agents dropping out in each step is equal, but it's not necessary that it's true. You can adjust it to 1 or 3 as per how your repository is behaving. And that offers realistic simulation and also offers stress testing because you will know what will happen in cases of high dropout and heavy task load by like increasing the number of dropouts. Similarly, the system scalability insights are observed and we have a balanced ecosystem modeling because of these 2 new effects, these 2 new things that I've added in the UI, and of course improved user engagement. So this is the previous UI where you can only adjust the number of steps and the number of agents. But here's the new AI with steps and agents remaining the same. We can adjust the tasks per step, and this offers to change the number of dropout agents per step, and we can run the simulation. So this is the output where I have set 3 tasks per step. So we can see 3 tasks given and 1 dropout agent. So this is the difference that I've observed in the resonance quotient. So the first graph that we see over here is the one where there are like 2 agents dropping out per step. So we see that it starts with 1 and it stays at 1 for like only 2 steps, and then it increases because of course it learns how it is supposed to react in a case. But then eventually when the agents keep dropping out, you are like, it's not a good state. So it drops down and it also goes down below 1. So it goes like it's somewhere between 0 to 1, so 0 point something. But if it's only 1 agent dropping out, the resonance quotient is around 1 in the beginning for the first 3 steps, and then it increases slowly. So it's gradual, but over here it's steep. And then we see that it drops, but then it does not go below 1. So yeah, it is definitely because of the dropout agents. So this is what I've observed. Also, I was asked to work on the visualization. So I worked on the visualization using NetLogo. So what is happening over here is that I actually don't have a video, but I'll upload the video through a drive link. I've uploaded a screenshot over here. So what is happening is that the blue circles that we see over here are the agents, and these yellow ones are the tasks. So for each step, these agents will move a bit, and then they will, when they touch the task, they will stop moving. So what happens is we are restricted over here, so we can't visualize for every single step because I'm using the NetLogo library and not the NetLogo simulation, not the NetLogo itself. So it's actually just a smaller version of it. So what is happening is that we cannot visualize every single step, but we can visualize it over 7 steps. So these, like, I think I set it around 40 to 50 agents. So these blue circles are those many agents, and in the whole simulation for that particular run, how many of our agents finish the number of tasks that they do, the circle reaches the square, yellow square, orange square, it touches it, that means it has done the task, and that's what happens. And then once the simulation is over, these stop moving and they become stable. So I was explaining why I couldn't use NetLogo, because NetLogo, the whole system itself requires a separate code, and shifting everything to that code, my entire simulation to that code is a bit hard at this stage. So I just used the NetLogo library, and I'm trying to do my best with it. So this is what I've achieved. And yeah, so I updated the README according to whatever I have done this week. and I had a week 10 blog. Also, I think my blog is also public. The mid evaluation blog, midterm evaluation blog is also posted on Medium now. So yeah, that's it for today.
All right, that looks great. Thank you.
Yeah.
Yeah, so I like the visualization, and yeah, PyNet logo is fine. Net logo standalone it's written in Pascal, I think, or something like that. So
Yeah,
that it takes that, but the visualization like this is perfectly fine, especially considering that I think a lot of people in past years have either used Pygame or PyNet logo, or a Mesa, which is Python based, so everything is Python based. That's fine. And I did receive your blog post, so we have that up on the blog now. If you go to medium.com or Rel Group, that's our blog, and Vidhi has she has her blog post published up there, which is just an overview of the project progress to this point.
Right. For the visualization video thing, I'll send you a drive link where you can probably access it.
Okay, that sounds good.
Okay.
Thank you. Lalith, are you ready to go?
Yeah. So before going to the updates, I just wanted to discuss. I found two interesting works in the hypergraph, but they're not exactly using hypergraph neural networks for C. elegans. They're using hypergraphs as a matching algorithm, to identify the nucleus and all. So they are building this nice visualizations of how exactly the neurons have been placed inside the C. elegans from the top to the bottom. They are trying to understand using the hypergraph. They are laying out all these, considering this is like the one node. So they are building the edges or the hyper edges based on that. So here we see the TR hole has the hyper edge, and so they have technically modeled properly using the hypergraph this work. So this was like an interesting read. I was able to understand the complete nuances of it, but I thought that this was one of the good line of work. And in the same line, the same first author, the author's name was Andrew. So the second the Andrew have published one more paper, but this is again the hypergraph tracking. So in the tracking, they have selected certain way of doing this mechanism. They have used proper neural networks, but not exactly hypergraph neural networks. They've used simple graph neural networks, and they use unit style architecture. This is simple unit style architecture. Where exactly? Yeah, so they have exactly used type to extract the volume. So they are able to identify these locations and then map it using cell nuclear detection. It was previous one of the works which was pushed into repo of cells segmentation SAM that we have already have. But there are no explicit works where they use hypergraph neural networks. So that's just one thing I just wanted to just discuss about.
Yeah.
So
So that first paper, that's in late embryogenesis. So let's look at that paper again.
Yeah. So I'll probably share these two links in the group.
Okay.
It will be useful. And so I was discussing about the visualization part. So after training neural network, it took me a little time to understand how do I have to visualize them. So I initially started off with the two-dimensional visualizations of the embeddings, how the network have been understood the entire data over a period of time. So for a one snapshot, this is how it understands. So when we move forward, when we want to understand, okay, suppose you want to cluster them together, saying that, per se, there are certain centriomes that have been formed, or, there are certain cells which actually have to come together. Then we can do a short period of clustering. And when we apply this clustering, we'll be able to see that, okay, so these two belongs to a common grain of clusters. These are like a confined batch of clusters. So this is something that we have to decide after seeing how the embeddings were saved. Later on in the step, I wanted to go into more details and see that, because this is not an more intuitive, because this is not a scalable plot. So when we have like more embeddings or we have more data, we will not be able to see this. Suppose we want like 7 or 8 or like 9 or 10 like visualizations, then this convex cell actually overlaps, so we couldn't make it. And in 3D we cannot show exactly how it's done. So I wanted to understand in how it looks, the plot that we were discussing last time is the star expansion. So in the star expansion, I was plotting how the things, the embeddings look like. So they're dividing in this certain fashion. So this now when we try to point it out on certain node, then it will give us the exact location, the cell ID, and also the lineage information. And the center, the squared box, I have represented as an hyper edge. So this is the edge which actually maps all these things together compactly. So this is a 2D representation, but we want to understand in a 3-dimensional level. So what I have ran a script is, so we have this nice 3-dimensional visualizations of the complete embeddings. So where exactly they were starting, where exactly they were ending. So these are the 3-dimensional embedding representations of the C elegans. What is the neural network that it has been learned underneath. So this is the representations it has done. So that we were able to understand. And if you want to, group them into certain number of clusters manually, so we can do that. We can apply a clustering algorithms like k-means, and we can cluster the group of, the nodes which actually come together, the hyper nodes, so that we can build a bunch of, a collector of graph. Say this is a whole, a group which actually behave in a similar pattern. So that's the thing that I was doing this week. It's, struggling for like 2 weeks probably, but yeah. So this is my side of the bit.
Great. Yeah, that's great. So the color bar is what? Like that's.
Yeah. Actually, this is little cluttered. I try to remove this part, but I'll try to, fix the things here.
So, but the different radio stars are different colors. What do those colors mean?
So what the colors actually mean that, they are a certain group. Suppose we say that we saw, in 2D, when we are trying to see this red, green, or yellow and violet, when we are trying to see these are like a certain type of groups. So we are manually grouping them. So based on a certain characteristics or behavior. So now we need to understand, suppose we cluster in certain manner. So we are trying to understand what is the group behavior. Suppose I say red, all, this red lines and blue nodes. So I wanted to understand what could be the group behavior. So based on the lineage information, based on the, the connectivity patterns. Suppose we say these are like the hyper edges. So then I wanted to understand that, like now I have given a color pattern, but in the long run we can use suppose like n number of clusters or n number of hyper nodes, hyper edges. So these edge carry the information about the group. It has a holistic understanding of the cells which are actually surrounded about, and lineage information it shares. So what does it mean? So in order to understand that aspect, suppose I point it out here, then I'll be able to get the lineage information. So from P0 it started to then AB, AB to ABP, ABP to ABL. That's how I will be able to see like where it has been transited. So I think this visualization is more intuitive when we try to understand more in detail. So it's like, suppose we group them into like 20 and say that this forms actually a centration. So then maybe we can manually justify that, okay, pointing out at this, so where it started and where it ended. So based on that, we'll have an intuition that, okay, so maybe there is a possibility of forming a centration, and this neural network has learned these embeddings in this certain manner. So that's what I understand. That's how I colored.
Okay, that's good. All right, thank you. And then going back to that. Oh, go ahead.
Also, I wanted to make a report so that I could detail everything into one place. So I was just thinking that so from week 1, I haven't made a crisp report. So I wanted to make it in a certain, proper journal format, because I didn't see any paper, or a type of work which was published under this, particular area. So maybe we can draft a paper and maybe we can refine it, iterating, discussing about like what things can be done in a good way. I think I need to start off with the literature and writing a little bit is what I believe. So is that
Okay.
so?
Sounds good. Why don't we go back to that first paper, the
This one.
2104? Yeah.
Should I share the screen? Am I sharing the screen?
Yeah, you're sharing the screen. Just kind of walk us through the paper, what they're doing.
Sure. Yeah. So the first, so they are like, the problem was like point set matching. So this is one of the standard problem in the computer vision what we see, and there are like certain point transformations that are actually used in different parts of the computer vision too. But what they wanted to see is, suppose we use that point set matching, and it actually requires like a higher characterization and underlying relationships. So they thought that in order to showcase these point sets, they wanted to use the hypergraphs as an extension to the traditional hypergraphs. So because the hypergraphs have like an instead of having a dyadic connection, they have a connection which actually groups a certain number of edges. So the existing hypergraph model point set matching methods, they have certain limitations. These people have used exact hypergraph matching, and so they have like a certain intuitive way of solving this. So first, what they have is this classical branch and bound paradigm, which is like this is quite an old method in the computer science where they try to understand graph matching. So, and what these people have tried is, so instead of using this old approach, they have tried this exact hypergraph matching approach, where they look this as like point matching problem. So when we go into the visualization, probably we can understand this. Yeah. So maybe this will give us a little understanding about the holistic picture. So here they manually identify the seam cell nuclei from the C elegans embryo. So the cell pairs form. So whatever the pairs, the cell form, they're labeled into like blue, red, and the green, and they have shown these connections how they are like connecting and all. So here they have the depth here is measured in like micrometers, and I think this was calcium imaging. I'm not really sure, but I think I've read about this. But yeah. So it's in just anterior posterior direction. It's in that orientation. And when they are trying to understand the seam cells, they see a certain posture. So this posture, or the topology, is actually captured using the hypergraphs. So they also say that they have this natural cubic splines. So here we see the splines that they draw in the figure A. So these are like cubic splines. So when they draw these cubic splines, they are able to see that the seam cells have been like coiled like a body. So because initially the cell is actually coiled, and then when we actually stretch it and we see it, so it actually looks like this. So hypergraph can actually help us to understand this, the pattern. And then when we fit these splines, the fit splines are used to untwist the worm and then generate the remapping strength and positions like in the right side. And so in order to track those points, how the worm has been there, and then they can project it back. So this is what they actually intuitively, say. And in abstract, they'll detail about this in more. So these C. elegans embryo can be used for cell tracking studies to understand how cell movements drive the development of specific embryo tissue. However, twitching due to muscular activity in the late stage of embryo is like invalidates the traditional cell tracking approach. So because when the twitching is happening, if you draw a linear edge, so what actually happens is instead of forming, so this is like a geodesic distance. So this geodesic distance, or like what is called, the curvature is not is absent. So in order to capture this curvature, probably they have used hypergraph as like an intuitive mechanism, and so they and to go more into details, yeah. So the embryo possesses a small set of cells which together act like a judicial markers to approximate the coiled embryo posture, serving as a frame of reference to track the cells of various cell tissues during the late stage embryogenesis. And so they say that the current approaches, they do not are like little time consuming and not accurate as what they have proposed. And this is more biologically inspired hypergraphical model, and it actually fits into a real problem like C. elegans. So this is what is their idea, and they talk little deeper into like the mathematics of hypergraphs. So how they have been done, and what is the existing literature, and why is it a problem which is not tractable, well, intractable. So it's more like that. I just gave a crisp read of the work, but I think a little deep could actually help us to, when we are like doing the literature survey, it will be great.
Yeah. So they're using the hyper nodes or hypergraphs to estimate
to do
from,
cell tracking.
Yeah. So I guess they would have like a population of candidate cells in a hyper node, and that would represent different positions, and then they just have to pick the right one. Okay.
And from that, they're able to draw these positions instead of having a straight one-to-one connection. They are connecting all those things using a hyper.
Okay. Yeah, that's pretty cool. All right. Let's see. We have. Yeah, and then the other paper, Layla showed the other paper, or shared the other paper as well. Okay. Well, that's good. Thank you for that update.
Yeah. I'll probably read into the literature more and come back. So, but I didn't see much of the papers. I saw only these two papers, that too with a very hard search. It took me a lot of time to find that papers too.
Like I said in Diva Worm last week, I'm actually surprised there was one paper on it.
Oh.
So, I mean, because I'm not aware of people doing this, but like, using it for like, because
so
a lot of the
like
cell tracking.
Computer vision task. So they're like trying to match using the data that's actually openly available. So it's not trying to understand the biological significance of how the worm grows, or the anastomosis happening, the cell tissue is forming. So they're not focusing on these parts. Right. So it's kind of unique approach, I believe.
Yeah. Thanks. All right, Jayadratha, are you ready to present your update?
Yeah. Okay. So last week I've shown some visualizations, and this time I have created one interactive visualization, and this is how development that is happening over time. But like it is taking some time for me. One thing I was missing was in this visualization is like here I have shown each kind of nodes that is neuron and its connection. So these are presynaptic neuron and this is postsynaptic neuron, and the connection is like between pre to post, and this is the connection strength, and this is a weighted graph and a directed graph. So pre to post there are some synapses, and there are different stages. So stage 1 to stage 8, that is what I have shown here. And different color signifies different type of nodes. So suppose this is sensory type of neuron, and this is muscle type of neuron, and this color signifies motor neuron, like that way. And these are the like connections here, and here for each node I have shown how many connections it has, and what stage it is now in. So suppose like this node is connected to 6 other neurons. So, and all 6 are chemical, and those chemical neurons, those are shown through this blue color edges, and this orange color edge, it shows the electrical kind of connection. And I also tried to show, like, once I hover on these edges, it should show which like its connection, and I mean how many synapses are there, and what are the like it is connected to whom. But I'm still like figuring it out how to add that, and the rest of the things I have done. So if I play, so it will show all 8 different timestamps. So till 8 timestamp, we can see the overall strength that is happening. That means brain is maturing over time, and it is also showing the like direction. So this is what I told I'll add this time, but this edges part I'll do maybe. I'm trying to do, but I'm getting some errors, so I'll show maybe next time. And the other thing is, so this is like how network that is developing over time, neuronal network of the C. elegans. And the other thing I wanted to show was DTDG, if it can predict some intermediate step. So I read few of the work and papers, and most I didn't find any paper that can show entire graph in the middle. Like there are 2 different timestamps, and there are in those 2 timestamps there are 2 different graphs, and in the middle, if I want to create one, I didn't find such like DTDG network available. So but what I found is like it is basically doing one task, that is most of the papers that is doing link prediction kind of tasks. So suppose I know like till 7 number of different timestamp, I know how the graph is evolving or the network is evolving. At 8th timestamp, if I want to know if there is any connection between this and this, so this node and this node, that is this neuron and this neuron, that it can predict with some kind of accuracy. But the problem is for C. elegans, it is already there, and researchers, they already have found it, and it is mostly deterministic, not like it will change. So then I thought maybe it has no use to use such network. If we can get something within intermediate steps, the entire graph, if we can generate, that would have been useful. But if there is any connection that can like happen in some like timestamp, is it valuable? So maybe it is not. So that is what like what I have read and understood this way.
Okay. That looks good. Yeah, thank you.
Thanks. Any feedback or anything?
Well, I think this is, great that you're kind of getting this sort of, formalized. So last week you showed these graphs, but you didn't have them labeled, or you didn't show the animation. I think the animation is interesting because you see the difference between the 2 time steps. Now, of course, you can't necessarily interpolate between them, because, it isn't necessarily just a smooth transition between the two time points. I mean, especially with synaptogenesis and that, there are fluctuations, and so we don't want to assume that there's this smooth transition. I mean, there is a deterministic component to this network, so it doesn't really change. But then the stuff that does change fluctuates, and we really, if we want to say anything about that, we would have to have data to back it up. So I think this is fine, just having like the transition points, seeing what changes, what doesn't, between the time points.
Okay.
Other than that, I don't know. Like, what are your steps like for next week?
Yeah, the thing is, I have not like pushed any code till now, so I'll probably clean everything what I have till now what I have done, and I'll maybe push some part of the like code to the repo, and also I'll start writing like readme file, and maybe I'll do some experiment on this visualization. Maybe I'll add few more just for completeness. This is what I want to do.
Okay.
Because rest of the thing, most of the thing I have done till this point. So we completed the cell lineage part, and also like how from birth to like I mean for different kind of uh, like cell divisions from birth time to up to some certain time, how the cell division is happening. Those visualization we have done, and also I have shown these for cell lineage how the network is evolving. Also, I have done some TGN kind of network I have shown, and for this cell lineage I have shown how continuous time dynamic graph is working there. Probably I'll add few more model, or maybe I'll try to improve the results there. There we got some good results, around 80% or so AUC. And now for this connectome, I don't think there is, I mean, as we have discussed now, there is no scope of using this TTDG model. So maybe I'll wrap this connectome part till this visualization thing, and maybe I'll write the report and those writing part, and nicely arrange whatever code we have done, and clean those code, and maybe put it in GitHub.
Okay, yeah, that would be great. Please update the repo this week.
Sure.
Yeah.
Sure.
All right, so I guess those are our updates from GSoC people. All right, so actually, Jesse posted. Let's take a look at this. This is Vidhi's, and the link is in the chat. This is Vidhi's post to our Medium, so congratulations, Vidhi. This is developing Sustain Hub, and this is her midterm report. So it's just, she's the author, and it's published on our lab blog. It's a Medium blog, so we have, for our REL group, we have, a pretty robust blog that, we always invite people to post on. Jesse's posted a lot on there. I've posted a lot on there. Various people have posted when they're in the lab. So if you're interested in posting to our lab Medium, please let us know.
Yeah, I just wanted to say hi. I wasn't able to join the GSoC meet the last 2 weeks. I think the timings weren't matching up. So yeah, last week after the SMN meet on Saturday, I kind of connected with Vidhi to kind of, just get an update. And then we had discussed some things as well, that some suggestions I had. So I think even today I joined like slightly late. So she's already finished her update, but I'll connect with her again as well. And yeah, just if anybody needs help writing on the Medium or like publishing to the Medium, so you can reach out to me as well since I've done it.
All right, thank you. Jess, did you have anything you wanted to update us on?
Jeez, about this week in general. Nothing. Nothing really is on the tip of my mind. There's a bunch of events and things happening upcoming that are maybe more to talk about Saturday. It's been I'm hoping something with data indirection will be accepted in certain places, and there's some local events. But also future plans to discuss as well, but those can be elsewhere. But yeah, just to double down on the writing and publishing stuff, I know it's sort of getting towards, I don't know, what is the actual end date for our summer this year? The summer session?
I think in early September. I'm not really sure yet. I
Yeah.
know the schedule's up, but yeah, we're going to be winding up in early fall, I think, these meetings.
Yeah, so I know it's kind of like final pushes and finding some documentation, maybe a final presentation or some final tying things together. But as we're doing that, and obviously Sarrah's done a lot, and it's a great resource and very helpful. So she's willing to help out with folks here, and we appreciate that very much. And if you have questions about, putting things in Medium, making a LinkedIn post, or how do you present yourself, your ideas and different things to the public, we can help with that. I think there's a lot of what I'm doing with some other folks and things too. So yeah, how do we, obviously first and foremost finishing up and making the most of your project and time here. So then thinking like sort of the, I don't want to say exit strategy, but just where do you go next with it? How do you make it something that other people can see, or that you feel good sharing after the fact? And we're not quite there yet, but just to note that you have support on that front too, as you link it. My only other, I guess, slight note is I'm looking forward to seeing Morgan in person later this week at an event here in Boston. I'm like 99% sure I'm going to be going to it, and hopefully some of what I've submitted will be accepted there too for GoPro and some data interaction stuff and other things. But yeah, that's it. There's other stuff that I may not have time to get into it right now, and I'm sure there's other things to talk about anyway. So I'll let it be on that front.
All right, that sounds good. Thank you. Let me share something with you. It's a feature. This week's feature is on taking open source software and building production-ready applications. If we go to our web, we go to our GitHub repository on the web, you'll see that we have, of course, a lot of different repositories, and I teach this course called Project Management. And I want to go over something that I have worked on in project management in these project management repositories. So every semester I teach this and I prepare a repository for this. And I have different demos in the repository. So this was from fall term. And I have this little demo of Dev v Prod. And so the significance of this demo is to show that when you work from GitHub, especially in an open source project, you usually have these different pipelines or workflows that you work from. So let's say we start our project and we start with, our team is going to divide up the tasks. and they want to do some experimentation, and they want to make maybe some pushes to GitHub to keep track of the different experiments, to keep track of the code, to share it with collaborators. Now that may be a little bit, you may not want to put that in the main branch, which is the default branch. So what you'll do is you'll create this branch, you'll engage in what they call branch development, and you'll create something called dev. And if you go to dev, you can have this branch which has different files, different commits than the main branch, but they're accessible within the same GitHub repository. So this is our main branch here. This has this README. It says, welcome to this dev v prod branch demo. Select the dev or prod branch to continue. So I go over here and I find my dev branch. And here I say this is the dev branch. So you see it's that README was, I committed that README to the main. And then I went to edit it and I changed the text in the README. And then I committed that not to main, but to dev. So these two READMEs are different. They're different commits. They have different information. And you can do this with your different branches. And it's very similar to forking something. So if I fork a repository into my other, my other account, if I have a different organization I want to fork something to, then I can do that, and I can have two different versions. In this case, I work from the same repository. I just have different branches managing different information. Okay, so that's dev, and of course dev is where you have, maybe you want to work out some ideas, maybe you want to work out some things like that. But then of course we have production, and so production is very different from development, and we want to work out different ideas within production. And so I want to create a new branch called prod, and this is prod, this is the prod branch. So again, I can take things from dev, I can recommit them on prod, I can push them into prod branch. That's the setup for this, and I want to make sure we understand what I'm talking about when I talk about putting things into production, the workflows and all that. So these are some articles talking about production. This is not like the mechanics of the GitHub pushes, but this is about these different, how do we put open source software into production? And as it turns out, it's much more complicated than just putting things into production from the dev branch. It's actually quite hard, as I found in researching this, to put things, open source software, into production and make them production ready. Because as you can imagine, in open source software, and then we have a lot of contributors maybe to dev, and we have to figure out which of those things to put into prod. So we're putting things into prod, and that's where we're going to take the things and put them into maybe production, into like a Streamlit app, or maybe like a Shiny app, or maybe some executable or some formal release. And so that's the problem that a lot of open source projects have. So this is the first article, 13 must-know open source software to build production-ready AI apps. So this is where, this is this person's Sunil Kumar Dash, and they're going to tell us a little bit about how, from their experience, what you need to do to put open source software into production. So a little bit about this article. I've been developing both AI and non-AI applications for some time now. While creating a prototype can be relatively straightforward, building AI systems that are truly ready for the real world is a much more challenging task. So the software needs to have, aside from this sort of branch development and this product management aspect, it needs to have other attributes as well. It needs to be reliable and well maintained. It needs to adhere to security standards, and it needs to be scalable, performant, fail-safe, and so on. So a lot of times when we're building open source software, and there is this perception that when you build open source software, you're flying by the seat of your pants. You're getting contributors, and they're putting things in, and it's this big pot that you have to kind of stir and make work. But actually, you can manage open source projects a little bit more effectively, especially in using these different this branch type development, these different branches, and not just prod and dev and main, but you can create things like security branches and other things, and then push those later to maybe to prod, and then put things in production from there. Actually, put things into production outside of GitHub. So there is this aspect of having to think about these other additional things. Reliability and maintenance, of course, in open source software, we do a lot of maintenance. We have to think about hiring maintainers and having a regularly scheduled update schedule. So like if we want to schedule new versions, we may do that every 3 months or every 4 months, keeping the software relevant, but also not, falling too far behind, and, being consistent. So this article continues, despite all the buzz around AI, the ecosystem for developing production-ready AI applications is still in its early stages. So this is where, this is specific to AI applications, and we want to be able to have these production-ready apps for AI. We can develop things on GitHub all we want, but to get them really in a position where we can put them out into the world and, have people use them seamlessly is maybe a bit it's a ways off. And so this is a list of open source software options to help you build production-ready AI applications. So you have Composio, which is seamless integration of tools with large language models, Weaviate, Haystack, and these are just frameworks and other types of ways to coordinate your tools to put them into production. So we're working from GitHub, but we also have these other tools that we need to bring to bear for these AI apps to get them to final production. So you can manage your code on GitHub, but you also need to integrate other tools into your workflow as well. And that might come post prod branch, post GitHub. And then you might maybe archive your version on GitHub, and you can put release versions in your GitHub repository. But a lot of this other stuff would take place outside of GitHub. So they kind of go through these tools and how to integrate large language models and integrate GitHub with large language models, and work from like APIs and things like that. And it kind of goes through some of these demos. And so I would leave that up to you to if you're interested in this, to look at how they structure this. You're building basically these different pipelines for putting things into production. So this is an article, 4 considerations when using open source in production. This is actually from 2010, I think. So this is a bit old, but it's still kind of is, I think, timeless advice. So this is just thinking about it from an IT perspective, an IT staff and developer perspective. So a lot of times, you think about your pipeline, you think about your GitHub project management techniques, you think about your sort of branch development structure, and then you think about the tools you need to integrate with that to put things into production. But there are these non-technical aspects that we need to think about as well. And so, a lot of times in IT teams, we think about these technical problems, we work out really good solutions, we work out really, seamless pipelines, and we have that all mastered, and we can actually put things into production, although it's sometimes very hard work. But then you have these non-technical issues you need to think about that will affect your production environment and your release. So the first thing is the license that you use. So you need to be able to know what it means when you use some software, or when your users use some software, or if you adopt a license, what that license entails. So the first step is understand all the licenses. When I say all the licenses, I mean that they refer to this idea of the license, but the license is It could be that you say adopt an MIT license for your project, for your repository, but there are dependencies that have different licenses. And you don't understand that at the time when you put it into production. And then that comes back to bite you when you have to maintain the software or update the software, or it just, you're not using the software in accordance with the underlying licenses. So it's important to deal with. Most people realize that they need to understand the license, in quotes, associated with a piece of open source software, but most people don't realize there's often more than one license associated with individual open source packages. So a lot of times you'll build upon open source packages, you'll work from those, but then they have their own licenses. Sometimes they're incompatible with the license you choose for your repository, so you need to understand how all that works. Open source packages often bundle other open source components, which may have different licenses. There are also many cases where a package includes specific files or pieces of code under different licenses. You need to find, review, and follow all of those bundled licenses. For example, if a project is licensed under the Apache license, but includes other open source code provided under a GPL license, you must comply with each of those licenses for the relevant portion of the software. So this means that, if I have an Apache license and a GPL license, and sometimes those are somewhat incompatible. So you need to figure out a strategy for satisfying all those constraints. The second part is vet the project and community, and this is important when you're dealing with adopting code from like an underlying package. So in the same way that you're dealing with the license, in quotes, you're dealing with the reputation of the project that you're drawing from. And this holds even if you're working from your own project. If you're working from your own project, you want to maintain a good reputation in the community, and more importantly, you want to put that out front in the repository. So a lot of repositories will have badges where they report sort of their relevance and their currency, how current they are with things, and other things that sort of advertise that we're not a dead project. So again, a lot of times you vet, proprietary vendors, and the same happens for open source projects. You want to understand, like, if the community is sustainable, if the community is active, if they've been around a long time, if they have like a plan for making regular releases, things like that. And so the more of these positive attributes they have, the more likely it is that they will be around for the long term, that you won't get stuck with legacy code or, things that can't be updated, or other problems that might crop up. And so it's not unusual to find code from dead projects backed by one uninterested developer in major open source repositories. So your repository might have some things in dev that are maybe dead software. We're in dev and we're adopting our packages and we don't really vet them properly. And we have dead pieces of software in dev. And then we go to build things in prod. And so we say, well, we're going to build things in prod. We're going to push things out. But we have dead packages that then propagate their way to prod and then propagate their way out the door to some final production pipeline and then out into a release. And then the problem with that is we can't maintain that very easily. So you may decide that you're comfortable using open source code from an inactive project and self-supporting it, but you'll want to make sure that you've made an informed decision. So it might mean that if I have dead code in dev, I could create another branch that's like, maintaining that code within our organization. It's perfectly fine to do, but you need to understand that. You need to know, you need to price that into what you're doing and invest the resources accordingly. Okay, so you just want to be able to assess that and not get caught without any options. So, and then there's some tools that allow you to sort of evaluate these. But really, I think it's just, having a set of criterion for your project or organization and then meeting those criteria. And then this third point, watch out for incidental distribution. So many IT organizations believe that the copyleft clauses of a license like GPL do not apply to them because they do not sell software. So copyleft is sort of the way that open source licenses are, the principle under which they work. Copyright, of course, is where you have restrictive terms for reusing content. So if I have some text on the web and I have a C with a circle around it, that means that you're not supposed to copy it and you're supposed to just read it and maybe pay for it if I charge you. Very restrictive and probably very impractical for the web. Copyleft, on the other hand, works that you can take that text, you can freely modify it, and then there are certain conditions that I give you for that reuse. And the same holds true for software. But sometimes people kind of assume that they can do whatever they want in a copyleft framework. So, if you're not doing anything proprietary, it might mean that, well, since I'm not trying to make money off of this, I can do whatever I want. And that's actually not what copyleft is about. It's about attribution. So a lot of times copyleft licenses will require things from, users, in this case, other organizations, that are non-monetary, like attribution or other types of obligations. And so this is very much, the whole idea of copyleft is very much it kind of fills in that gap between the proprietary world and having chaos within the non-proprietary world. So it's something that we have to abide by and understand the conditions of. So it's highly likely that some of the applications you develop are distributed outside the walls of your organization. It might be an iPhone app or your corporate website, tools for partners, or software that's embedded in a product you give to customers. And so you can't just violate the terms of your license just because you're doing things that are not proprietary. Okay. And then 4 is don't forget about support. So for any open source software going into production, you'll need to consider the level of support that's needed. In the case of small libraries where you have extensive in-house expertise, self-support using community mailing lists is a reasonable option. If you don't have internal expertise or you need help with more complex and critical systems, then you may want to consider other support options. So there are a lot of ways you can offer support for what you're doing. And again, this can all be managed within a repository within your branch development scheme. So you might have like a maintenance branch where you're taking code from dev branch or prod branch and you're evaluating it on a regular basis. So you might have a schedule where you say, we're going to go to dev and we're going to make sure that all this code is maintained well, that it meets security standards. And we might have a plan for testing the code, unit testing the code, say, where we can make sure that it's okay, every 3 months. And then when we have something there in dev like that, or in prod, we can be assured that when we put that into actual production, we can then have something that's reliable. We also can do maintenance through pull requests, or we can do maintenance through sort of evaluating what's in main. So sometimes in main, we'll do maintenance related tests. Okay, so then the last article here is this article on why open source is a terrible way to build products. Yeah, most great products use open source. So this is from Glauber Costa. This is from 2023. It says, open source is eating the world, but is it the best way to build products? And so, there's this thinking that goes something like this. We use open source. It's great for kind of developing things, bootstrapping things from nothing. But at the same time, in terms of product development, product management, it really doesn't, it doesn't match. And I think mainly for the reasons I mentioned. This is a little anecdote, though. Some 15 years ago, I had a falling out with an acquaintance of mine over a realization I had that open source is an absolutely terrible way to build products. When I shared this, I could immediately feel the atmosphere in the room darkening. I did say that I thought open source was a great way to build technology, but that didn't do much to temper the situation. So they were both very active in open source, and so they had drunken the Kool-Aid, so to speak, and they were angry, or the other person was angry because, there's this aspect of sort of cult-like behavior in technology that if you're really into something and you kind of criticize it, that you're really kind of betraying the thing that you're interested in. So maybe that's what part of this was. But it's a good point, because you have to think about production, and you have to think about innovation, and you have to think about, kind of developing things all separately. They don't all go together. One doesn't presume the other. That maybe open source wasn't a great way to build products was an early realization of mine at the time. But 15 years and 2 startups later, I am now even more confident in my conclusion than before. That is, while open source is a great way to build technology, it is an absolutely disastrous way to build a product. But I've also come to the paradoxical conclusion that this is exactly why open source should be a part of every product pipeline. So I guess the idea here is that open source is a good way to get to the product, but then building the product maybe into production, putting it into production, maybe that should not be open source. And I guess the point here is that there are different ways you can apply open source to your project, like we talked about with, in a previous feature, we talked about how not every part of your project needs to be open source. You can have proprietary components. Likewise, you can have components where there's no open source part of it. In other words, you might have contributors in the dev and part of the pipeline. You might have maintainers, but the production part of the project or the organization is all sort of not proprietary, but it involves like a team that doesn't do open source like things. And so then again though, you have to think about products differently than you think about projects where you're getting contributors and all this. You have to think about what is a product and what is technology, and you have to make this distinction between a product and technology. So technological development is different from product development. So you take ChatGPT. This is a product based on the GPT-3 or 4 technology. So you have this technology, this framework, and you build products from it. This touches on the core of the issue. A product is created using technology and is a derivative work. Because we install those things like Linux or databases like Postgres, they have logos, mascots, conferences. We may think of them as products. But products also solve a particular problem or set of problems, have a particular form factor, warranties, guarantees, the supply chain, all of that. So open source products might try to make themselves useful for a wide audience and approximate products. But at the end of the day, they are not products. They're technologies, and they can't be products until you make a specific targeted product. And so it kind of goes through what makes a good technology. There are a number of different building blocks like affordability, versatility, adaptability. But then what makes a good product? That's something different. That's actually about marketing. It's about learning where your product fits, your use cases, your niche, and how you can deal with the sort of marketing of a specific thing that's a derivative of that technology. And so you can think about games in this way. You can build a game engine, and that can be open source, but then you want to put into production specific games. Well, those specific games are going to be the products, and those can't be developed in the same way as the platform. You have to have like marketing, you have to have thought go into like the actual market, the actual use cases, and so forth. And so this leads us to this point here. Open source makes for bad products. So it's not inherently impossible to build an open source product, but building a product using open source conveys no inherent advantage. You still need a strong vision and the determination to cut through the forest at a single place, foregoing every other alternative. And so, we want to be able to build things using open source software, have good open source communities, but you have to keep in mind that product is a separate layer, or a product is a separate layer, and it's very different from the actual thing that you're doing in your project or your repo. Okay, so that's all I had for that. Now, before we go, I don't know if Morgan had an update for us. If I don't know, he was on the bus before, so.
Oh, hi. Yeah, so like Jess said, there's a big hackathon this weekend locally here in San Francisco. I'm definitely trying to somewhat align it with OgHack in Boston, and looking with an eye to the Human Augmentation Summit on August 23rd. I've just been really enjoying the Beacon talks that I can catch. We're teaching people how to convert 3D printers into open source 3D bioprinters this weekend with Nucleate Dojo House. I will try and post more links and things to the kind of stuff that we're working on here. But been mostly busy. Oh, and Robot Fight Club tonight here in the basement of Frontier Tower.
Great.
Which is bringing in lots of people to the tower. It's more a spectacle than than kind of tech demo, but anyway, we'll try and post updates.
All right, sounds great. Yeah, I'd like to see some pictures of that. But...
Sure. yeah, we'll do.
All right, well, I think that's it for today. Thanks for attending, and see everyone next week.
Take care.
Take care.
Thank you.
Thank you.
Bye. See you.
Thank you.
See you.
Bye.
Bye.